really hard questions. I know. Einstein said if you can't explain it to probably don't fully understand it yourself.
Awesome. Good morning, everyone. So today's seminar speaker is Dr. Luke Mckay. He is an assistant research professor at the Department of Land Resources and Environmental Science, as well as the Center for Biofilm Engineering at Montana State University. He's a microbial ecologist and an astrobiologist who studies microorganisms living in extreme environments all over the globe, including Yellowstone National Park. And if he looks familiar, that's because he's one of the stars of the Netflix original documentary, The Most Unknown. And this was screened earlier this year at P Street. So I figure I would get him in to speak about his research to you all. He received a Bachelor of Science in Molecular Biology with a minor in chemistry at the University of Alabama at Birmingham. He then moved on to UNC's Chapel Hill campus, where he received both his master's and his PhD in marine sciences, examining microbial communities in hydrothermal systems. And then he moved on to do a postdoc at Montana State, where he is currently. And I believe today Luke is going to share some new exciting results that he's really thrilled to share with you today about Yellowstone National Park.
Thanks, Joy. Can everybody hear me? Okay, great. Title of my talk today is Early Evolved Microbial Metabolisms and Virus Host Dynamics Mediate Methane and Sulfur Cycles. That was already a really long title, but the ellipses after that, it should say in thermal environments, because that's really what I'm studying. So I chose to use this scanning electron micrograph as like the background for the title slide. This is a hydrothermal vent community of organisms in the bottom of Yellowstone Lake. And I'm going to talk more about that kind of the second half of my talk. But it sort of gives this, it portrays the idea in environmental microbiology as you just have this mixed up pasta of life, and you're trying to figure out who it all is and what are they doing, how are they linked to localized geochemistry, how are they modifying that. And so that's kind of every new environment I go to, it's always this. It's like, who's there, what are they doing, and how are they interacting with the geochem? I like to start with acknowledgements so I have enough time. I work a lot with Dr. Bill Inskeep and Matthew Fields at MSU, as well as Roland Hotspickler, Mincer Dlakic, and Corey is an undergrad who does a lot of the grunt work. Or did a lot of the grunt work in the first half of my talk. And then some collaborators from elsewhere, Dr. Olivia Nigro from Hawaii Pacific University. She's an amazing viral ecologist. And as you'll see throughout my talk, I'm starting to get very interested in viral ecology and how that relates to that pasta network of organisms. And Dr. Karen Luttrell at LSU, who is kind of further outside of my discipline, probably more in your discipline, volcanism and sort of geophysics, and she got us some really awesome samples to work with. So I threw her up here. Okay, so I know this isn't like a microbiology focused department, and so I wanted to start with some background info. This is a phylogenetic tree. And I'm going to show a few of these in the talk today, so I thought I'd just start with like a quick explanation. This is a comparison of DNA and DNA sequences from different organisms. And when two branches are close together on a phylogenetic tree, that means the DNA of those two organisms is quite similar. And then if two branches are very far apart, the mathematical difference between those DNA sequences is quite large. Okay, so branches close together are closely related. It's kind of a no-brainer. So this is our current most up-to-date understanding of the overall diversity of life on planet Earth. Okay, so every taxonomic lineage of life is that we know of so far, which we're constantly discovering new ones, but that we know of so far is on this tree. And so just to kind of orient you with what we're dealing with, here's human beings. This is Beyonce. Here's a Siberian tiger, a cockroach, a sea anemone, an oak tree, a radish, a daffodil, a portobello mushroom. That, I think, is Giardia. That is, it's Naglaria fowleri, a brain-eating amoeba. And then this one is Trichodesmus vaginalis. It's a very common parasite for humans. But the point being that when we think of diversity, of living diversity, like so much of the diversity that we're actually thinking of is captured within this tiny red circle on the overall tree of life, okay? And that circle is one of the 3 domains of life called eukarya. Now you also have the archaea, which is much larger than that over here, and then you have the bacteria, which is the big one. So those are the 3 domains of life. And so within the microbial side, the archaea and the bacteria, you have interesting organisms like this methanogen, Methanothermobacter thermatotrophicus. Then you have, I just want to back up real quick and say, everything in the eukarya, this tree doesn't just represent taxonomic diversity, but you can think about it in terms of metabolic diversity. So how are these organisms living? And everything in the red circle primarily gets its energy and carbon one of 2 ways. So it's either an oxygenic photosynthetic organism, like a plant, or it's an aerobic heterotroph. So it consumes organic carbon by respiring oxygen. So those are, and there is some anaerobe stuff going on here too, but for the most part, those are the 2 things that are happening in the red circle. Whereas once you move into the microbial world, you have, for instance, this organism that's taking carbon dioxide and hydrogen and making methane. This is a sulfide oxidizing organism. This is ammonium oxidizing organism. And this respires organic matter with oxidized iron. So it's respiring metal, basically. So this kind of establishes we have this very huge taxonomic DNA-based diversity, but also this massive, vast metabolic diversity as well in the microbial world. I just want to draw your attention real quick to this very large branch of the tree. This is called the candidate phyla radiation, and this was discovered 5 years ago. This entire section of the tree. So that's almost 1/3 of the known diversity of life on Earth was discovered 5 years ago. 6 years ago, we did not know that that branch was there. And so we're still trying to figure out what in the world those are doing. And that's a lot harder than it sounds. Okay, so environmental microbiology as a discipline, microbial ecology, is tasked with the question, okay, what is this part of the tree doing? Which is not easy. And so we're developing new techniques all the time to try and figure that kind of stuff out. Just want to see. Oh, here's a clock. Okay, perfect. Historically, microbiology has been based on cultivation. And we're in this era of microbiology where we're not throwing away the important information that we know from cultivation, but we're moving towards other techniques that can give us more information about the reality of environmentally, ecologically relevant populations. And so here you have a plate where someone's growing a monoculture. In other words, like everything on that plate is the same exact organism. It has one genome, it's doing one thing. And then you move towards high throughput sequencing where you go to an environment, it could literally be the carpet, and you take a swab, you extract all the DNA from all the microbes living in this section of carpet here, and you sequence all of that DNA together. And every single teeny tiny dot in this plot is a strand of DNA, and this is a plot showing all of the populations that are in that environment. And so we can really use high-throughput sequencing to understand full communities and also how individuals are acting within a community. So this is a lot of the kind of work that I do. Okay. And so moving on, that's the introduction. Now we're getting into the actual research. So I'm most interested in microorganisms, microbial populations that are involved in metabolisms based on sulfur or methane. And the reason for that is there's several lines of evidence that suggest that sulfur and methane-based metabolisms are probably some of the earliest evolved microbial ways of life, or ways of life, period. From phylogenetic trees where the deeply rooted organisms that anchor to the deepest points in the phylogenetic tree of life are oftentimes based on methane or sulfur metabolisms. If you do ancestral reconstructions of ancient metabolisms, you often converge on sulfur or methane-based metabolisms. And finally, the kind of nail in the coffin, which I'm sure a lot of you all are familiar with, is sort of isotopic evidence in the rock record of either light carbon or light sulfur or methane inclusions in rocks. And so these kinds of things would give us this suggestion that these are ancient ways of life. Okay, so that's kind of, now I go out into environments where these metabolisms take place. And I try and study these to understand how they're interacting with their environment. And so thinking about, this is the way that I think about this. So you have, in any given environment, you have a variety of single carbon compounds that can be involved in methane-based metabolisms, either the production of methane as different substrates for methane production, or methane can then be oxidized by some organisms back to other C1 compounds. And those are represented here. So you have all of these different, tons of different microbial transformations, conversions of these C1 compounds cycling through. And these are, the names of the different types of microbial metabolisms and also the enzymes that are responsible for that process, so we can look for those enzymes to understand that process. And as you can see, you've got the oxidation states listed over here for carbon in these compounds. And all of these conversions are, redox exchanges. So as these compounds as C1 compounds are being cycled by microorganisms, there's electrons moving around a lot in these systems. And so you can think about that as kind of forming this electron pool that then can be used by different types of metabolisms. Like over here I have all the redox exchanges happening during sulfur transformations. And so we are thinking about, in our systems, in our sort of high temperature geothermal, hydrothermal systems, how are these different types of metabolisms linked together, either directly in that it's actually a unified metabolism that an organism has, or a community level where there's a bunch of organisms doing this and a bunch of different organisms doing this, but electrons are being passed back and forth. So linkages between these early evolved metabolisms. Yes?
How can you tell whether the system you're looking at is an ancient one or something that's evolved more recently?
Well, there's a few different ways. So take methanogenesis, for example. Methanogenesis, there's a lot of diversity in methanogenesis as a metabolism, and there's much more recently evolved methanogens, and there's much more ancient evolved methanogens. And in a minute, I'm going to talk about how we figured out that one particular organism has probably a very ancient derived enzymatic complex for that process of methanogenesis. So I'll let you know when I'm at that slide, if that's cool. Okay, and so then not only that, I'm mostly focusing on suboxic environments, but oftentimes gradient environments. So where you're transitioning from a suboxic, the same community is existing across something suboxic and then also fully oxygenated. And so we think about that. It's not perfect, but it kind of sheds some light on the way we think, what happened to all the anaerobic metabolisms, all the sulfur and methane-based metabolisms through this transition of Earth's history with the oxygenation of the atmosphere, and then obviously changing the redox states of so many compounds involved in these processes. Okay, so this is kind of a summary of what I just said. All these red lines are microbial conversions of these carbon substrates or sulfur substrates, and they're potentially linked. And then just to make it more complicated, just for fun, it turns out that viruses actually affect the microbial communities in major ways. And most people think about viruses in terms of infection, lysis, that kind of thing. But we're coming to find out recently that viruses also carry auxiliary metabolic genes. So they might actually be able to encode enzymes themselves for certain processes that directly affect these cycles. And so that's one thing that we're getting really interested in our work. Okay, so finally we've gotten to Yellowstone National Park. Most of my work right now is in Yellowstone National Park. This is a wonderful place. If you've never been there, you should go. And Yellowstone is a supervolcano. This is the most recent caldera. And these are the two sites I'm going to talk about today in this talk. So I'm going to talk about a terrestrial geothermal hot spring, Washburn Hot Springs, and then I'm going to talk about a sublacustrine hydrothermal vent in the bottom of Yellowstone Lake. So first we'll talk about the hot spring, Washburn Hot Springs. And yeah, so the big overarching question, can we find evidence of ancient metabolisms in modern environments? You know, how do you make any sort of comment on what's early evolved or ancient based on extant metabolisms in life? So here is a close-up of Washburn. If you have been to Yellowstone, this is the canyon area. This is the Yellowstone River. And so this is the famous Yellowstone Falls right there at the beginning of this canyon. And this is Washburn Hot Springs here. So you hike along this trail back this way, and then you kind of cut through these fields and go back there. It's about 10 miles round trip hike. And that's what Washburn Hot Springs looks like. And we will go hike out there and take samples of the hot springs. And it's imperative that I say at this moment that this research was conducted under Yellowstone Research Permit, that number there. And no one should go and take samples unless you're authorized by the National Park Service, which we have permitted access to do. I actually am obligated to say that, so sorry for that brief little interlude. Okay, so here's Washburn Hot Springs. We hike up there and we take some samples. And this is sort of like the physicochemical characteristics. You have temperature between 65 and 70 Celsius. This is a bubbling hot spring, and so we take literally like a painter's pole with a huge beaker on it, and the beaker's upside down. It's about this big, and we drill a hole in the back of the beaker, put a little septum in it, stick this under the water, and then hover it above the bubbles. The bubbles fill up the beaker and then bring it closer to shore, and you can just use a syringe and needle and sample the gases out of the back of the beaker. And so we call those the source gases of this hot spring. So this is the composition of the source gases. As you can see, it's primarily carbon dioxide, methane, and hydrogen. 92% carbon dioxide, but a lot of methane, a lot of hydrogen. This is a very interesting hot spring. Sulfitic, it's also one of the stinkier ones. And pyritic, it's this dark, just like you see here in this Google Maps image. It's very dark. And suboxic, obviously undetectable oxygen. And near marine levels of sulfate, 22 millimolar ammonium sulfate. So it's a very interesting system that is not well represented anywhere else in the world. And so I kind of know this because I know what's coming, and that's that we discovered all sorts of new interesting life going on in this hot spring. So this is what we do. We go and we take a sample of mud in a falcon tube, bring it back to the lab, and you have a rock star undergraduate do a DNA extraction on it, which involves putting the mud in a tube, adding glass beads. You shake the glass beads with the mud really fast. The glass beads break open all the cells that live inside. The microbial cells are lysed. They break open. You do a chemical extraction and purify the DNA. You send your DNA to a sequencing facility. A sequencing facility basically converts your molecular DNA to a computer file. So you have all these sequences. And then you have all these little short reads from this mixed community of organisms, from a bunch of different types of organisms doing different things. And you assemble those, and you get, based on overlapping reads, and then you get these longer things we call scaffolds. But still, that's all mixed up. And one of the coolest things you can do, and this is basically the field of metagenomics, this is one of the things it's based on, is the ratio of the nucleotides, A to T to C to G, actually is constrained with the, each species will have a unique frequency of what we call a tetranucleotide frequency. And so you can plot those tetranucleotide frequencies. You take all these bits of DNA and you plot them. This is T stochastic neighbor embedding plot of nucleotide frequencies. And every single tiny dot is a strand of DNA, but a cluster that's the same color is strands of DNA that have the same tetranucleotide frequency. And so arguably, those clusters that are the same color are a unique population of microorganisms. And by population, I mean species, basically. So you have, this is the community in Washburn Hot Springs, and each one of these different colored clusters is a unique population, one constrained microbial genome recovered from the metagenome. Okay, so then you can focus in on one of those. You can find out all the different genes that belong in that genome, and you can reconstruct a metabolism from that organism. You can do this on a community level and an individual level. Yep.
So you've got like red dots on there,
No, it's just there's too many colors. So yeah, it's just reusing red, yeah. So just like your eyes would naturally say, this red cluster is one population and this is a different one. And this is also actually a two-dimensional screenshot of a three-dimensional display. So some of these are in front of other ones, which actually you can resolve with the 3 dimensions.
what's the axis on that plot? Or axis on that plot, or just dispersed?
I honestly, I'm not good at this part. It's called T-stochastic neighbor embedding, and it's kind of like principal coordinates, where it's collapsing multiple dimensions into fewer dimensions. But I can't really, I don't know enough about that actual technique. But I know that it works well for extracting microbial populations. The proof's in the pudding, because once you do this, you can double check by looking for the right genes you would expect to find in a genome. Okay, so in Washburn Hot Springs, which remember this whole thing was basically at Washburn Hot Springs, right? We went through this whole process. We are interested in methane metabolism. So we also, at the same time, we designed a primer set that targets the marker gene for methanogenesis or methanotrophy. It's the same gene that does both. And this is, again, another phylogenetic tree. And this is, again, 2 branches that are close together. That's very similar sequence. 2 branches that are far apart. It's very divergent, distant. And so this is the MCRA tree. That's the marker gene for methanogenesis. So we're only comparing the one gene that every organism that is involved in methane metabolism uses. And a lot of developments have happened here in recent years, and we were actually able to discover these by designing new primers for this gene and amplifying it. We were able to discover 2 new groups from Yellowstone to add to this tree. But what was really interesting was this core archaeota up here. We realized that this gene, no one's ever, core archaeota is an archaeal phylum level lineage, and a phylum is huge, right? Our phylum is chordata. So that's notochord, nerve cord, pharyngeal slits, and a tailbone. And so anything with that, including like a trout, is in our phylum. So it's a very broad taxonomic group. But this is a very broad taxonomic group of archaea that's very poorly understood. And we noticed that it had the gene for methane metabolism, and no one had ever shown that before. And methane metabolism was thought to be constrained to only these other groups of archaea. And so we were able to demonstrate that, oh, wow, this new lineage, deeply rooted in the tree of life, is able to conduct methane metabolism. And we were able to cross-reference this gene-based approach with this genome-based approach and find this gene in this genome and then examine that genome more closely. So instead of just looking at one gene, now we can relate it to all the other genes in the genome and see what else it might be doing. So Dr. Dlockick is an amazing bioinformatician at Montana State, and he did a lot of this work. This is just sort of an archaeal tree, a phylogenetic tree with a bacterial outgroup showing the position of this unknown core archaeota group. And it's a deeply rooted lineage in the archaea. And then, as I mentioned, and so prior to this, these are all the organisms that had methane-based metabolism in the tree of life. And so that's kind of the distribution of methane metabolism in the tree. And then likewise, these are all the organisms that had anaerobic sulfur-based metabolism. So dissimilatory sulfur metabolism in anoxic environments. And what we found in this organism is that it actually has the full enzymatic machinery. It can encode the full enzymatic machinery for both of these processes, which no one had ever discovered before. So this was a very, very exciting finding for us. And it prompted some big questions. So can these metabolisms actually interact with one another in a unified organism? But this is now getting to your question. So you can take a phylogeny of an enzyme or a gene, and you can do what's called an ancestral reconstruction. So you take all of the modern extant versions of that gene, and you basically use those in a model that builds the ancestral version of that gene. And then you can take that gene, and you can compare it back to all of the modern genes. And the one that's the modern gene, the modern day extant gene that is most closely related to your ancestral reconstruction is most likely the gene that is ancestral in that tree, if that makes sense. That's exactly what we did, and we found that so not only does this novel population of a deeply rooted archaeon have both genes for methane and sulfur metabolism, the gene for methane metabolism is the most closely related to the ancestral reconstruction. There's one question in the back real quick. I'm doing this in a mixed population, and we are actually now based on, I'm not going to talk about this actually, so it's good that you asked this, but we predicted what this organism is doing based on its genome assembled, or metagenome assembled genome, which is what we call when you extract an individual genome from a community of genomes. We predicted its metabolism, and now we have a colleague in Roland Hotzenpickler's lab at MSU who has used that prediction, and she has an active enrichment of this organism in the lab. So we haven't isolated it, but we have an enrichment, and we're very excited about that because this is really when the hypothesis testing happens. So, yeah. Quite confident, yeah, actually. So one thing, you can do a lot of bioinformatic techniques to sort of test the how good your bins are, your clusters of mixed sequence. Like, for example, organisms have what we call single copy genes, where it's a very important gene, but it's only got one copy of that. And so what you do to estimate the completeness of the genome you recover is you do a search, a hidden Markov model scan for all of the single copy genes known to belong to archaea or bacteria, depending on what you're looking at. And so we got an estimated completeness based on that of 95% complete genome of this one. But then you can also use that same technique to look for redundancy. So where do you find actually two versions of a single copy gene? Because that might indicate you have some level of contamination in your sequence. And we had about, I think it's 95% complete, 4% redundant, and a highly constrained tetranucleotide cluster. Yes, yes, very low. That is considered a high-quality MAG by metagenomic scientists.
So, to what extent is horizontal transfer affect these particular enzymes, and can you be sure you're not seeing that lateral transfer?
It's almost as if you know what I'm about to say next. So, what turns out is that while this gene appears to be ancestral, the methane metabolism gene, The gene for sulfur metabolism appears to have been horizontally acquired by this organism. And that's because we did the same ancestral reconstruction, but also if you look right here, these are all archaea, and they fall within a very huge cluster of bacteria. So this is bacteria, 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 and these right here, where this gene are archaeal. And so it could be a relic of the fact that we actually don't know a ton yet, and we're making new discoveries all the time. And indeed, in this very paper, we added additions to this tree. So 10 years from now, maybe those bacteria will be in a broader archaeal group. So horizontal gene transfer is actually very hard to constrain. But this would suggest at this point in time that the sulfur metabolism gene was horizontally acquired from Firmicutes bacteria later in the evolution of this organism. Okay, so because this organism has the gene set for methane metabolism and the gene set for sulfur metabolism, we were able to kind of use the other genes involved or in this genome to say, okay, well maybe this organism is a methanogen that has genes for reducing methanol with hydrogen as well. So it could be doing that. Or if you separately look at just the sulfur genes, maybe it's a sulfite-reducing organism with hydrogen. It has all the genes to do that. But what if these enzymatic systems actually interact with one another? So we predicted a brand new metabolism. This is a completely hypothetical. It's probably the most hypothetical thing that's ever been published in Nature Micro. But it's basically saying, this has never been established in a laboratory anywhere. But there are certain things you can predict based on interactions of different enzymes and homologies of different subunits or enzymes that are involved in methane metabolism and sulfur metabolism that actually have maybe a shared evolutionary history, like heterodisulfide reductases. So we were able to predict that maybe it actually can oxidize methane to methanol with sulfite in a previously unreported metabolism, which kind of gives it lends credence to this an idea of early life based on methane, which some folks have proposed, because methane would be a great source of energy and carbon at the same time. Okay, so, and I should mention, because this is really cool, I think, that it doesn't actually stop here. Here's the sort of reconstruction of that last metabolism where it's taking methane and it's oxidizing it to methanol, but actually has genes to take methanol and assimilate that as a carbon source. And that's really cool because methane, as everyone knows, is a very potent greenhouse gas. And so this organism, instead of oxidizing it to carbon dioxide, which is what every methane oxidizing organism usually does, this organism can potentially sequester carbon from methane into biomass, which is kind of a cool little side note in terms of this story. Okay, so finally with this very interesting organism, we decided that was very cool, but we took it a step further and we noticed that there is evidence of extreme viral pressure on this particular organism in this hot spring. And I don't mean to insult anyone's intelligence. I'm sure most of you know this, but CRISPR, clustered regularly interspaced palindromic repeats, most people who have heard of CRISPR think of it as a new technology for gene editing where you can very rapidly and efficiently edit genes. But this was invented long, long ago in the microbial world as a defense system against viral pressure. So just like humans are infected by the flu or whatever, microbes get viruses too, and they have a defense system called CRISPR that can recognize viral sequences and then chop it up, basically, chop up a viral genome. So what you can do is you can take the CRISPR defense system in the microbial genome and you can use that to find the actual viruses that are attacking that organism in the overall metagenome. And then you can look at those viral genomes and you can say, what are they doing? And so these particular viruses, among many other things, I just want to point out that they have some sulfur metabolism genes. So they do have AMGs, auxiliary metabolic genes, that they're encoding as part of the viral community. So they are probably also influencing this organism's ability to metabolize either by supplementation or by viral pressure, making it harder on the organism to influence the sulfur cycle at Washburn. Okay, so that's the end of the terrestrial hot spring story in this new population. Now we're going to move to a hydrothermal vent in Yellowstone Lake. We call this the deep hole. Fun fact about Yellowstone, the hottest fluid expression in Yellowstone National Park is in the bottom of Yellowstone Lake. It's none of the hot springs or geysers that you've heard of. Does anyone have a guess as to why that is? Pressure, exactly. So the boiling point at the bottom of the lake is 170 degrees Celsius of water. And so we've measured vents in the bottom of the deep hole, which is, I think, 130 meters, ejecting fluids at 150 C, and that's what we're going to talk about. Whereas terrestrial hot springs in the park, they top out at 90 degrees C because it's a high alpine system. There's actually less pressure at the surface. Okay, so our big question here, how do microorganisms influence the sulfur cycle at these hydrothermal vents? So here's Captain Dave Lavalvo on research vessel Annie, and here's the remotely operated vehicle Yogi going down to the bottom of the lake. Here's the lake, and right there is the deep hole off Stevenson Island. Here's a close-up of that deep hole. This is the deepest spot in the lake, and there are many different hydrothermal vents down here in the deep hole, and this is what they look like. They're not giant, black smokers like you might imagine in the ocean, but they're these small punctures, and there's very hot fluids, 150 C, coming out of these, and there's these microbial streamer communities that are attached to the vents. And that's what we're interested in studying, is these sort of large streamer communities that are waving around right there in that super hot fluid. Sulfur streamers in Yellowstone are nothing new. This is a video from Mammoth Hot Springs where the same types of populations exist. So they exist at Mammoth Hot Springs and at the bottom of the lake, but they're slightly different based on different chemistry and organisms present. But they're dominated by an organism called Sulfuri hydrogenebium. That's pretty much the white stringy material, but there's a lot of other organisms entangled. So there's a relative low diversity. That's what Yellowstone is really cool as coming from a microbiology perspective, because it's kind of like a nice natural laboratory. It has some amount of sort of controlled experimentation feel to it when cultivation in a lab is not possible. And so these organisms colonize these expressions, and they're relying on hydrogen sulfide that is coming out in the fluids. So here we've arrived again at this picture, which is now a hydrothermal vent streamer from Yellowstone Lake, and these are all the organisms entangled within that white streamer material. And Bill took this image. He loves doing electron microscopy, and he's very good at it. But what you see here is this incredible morphological diversity, thin filaments, large filaments, circular cocci, large, small. Here you have a diatom over here, which is probably not existing there, not living there, but it was sedimented down. And then another very, very tiny filament up there. So you have this interesting mixed community of organisms. About 50% archaea, 50% bacteria involved in that population. This is another tree, and just showing the dominant groups with the colored circles here. You don't have to pay attention to that. Sorry, I probably shouldn't have showed it. We did elemental analysis on these streamer materials, and we actually were able to discover that the largest filaments, microbial filaments, are accumulating elemental sulfur in their periplasm, kind of as like an electron donor reserve, which is pretty cool. So that was an interesting finding here. That's sulfur in blue, and you can see these little globules. We are able to predict, these are all sorts of techniques you can do when you're not able to isolate something in the lab and you're thinking about things on the community level. And you can use amino acid frequencies, specifically those 7 amino acids over the total amino acid content. That correlates very strongly, very well with optimal growth temperature. And so what we realized is that the populations that were present in this community actually sort of span the gamut from psychrophilic, cold-loving life to hyperthermophilic, very hot-loving, high-temperature-loving life. And the reason for that is because these are gradient organisms. So the temperature of the lake water is 8 degrees Celsius, and the temperature of the vent fluids is 150 degrees Celsius. So those streamers that are waving around at the bottom of the lake are waving around in a very severe thermal gradient. And it's also a sulfide to oxygen gradient, because the oxygenated lake water to highly sulfidic reduced vent fluids. And so this is a gradient in many ways. And our big question here is, okay, so you have this high source of hydrogen sulfide coming out in this vent, and then these are microbial conversions in the sulfur cycle. So these are the arrows here represent actual microbial enzymes that can make these conversions happen in the sulfur cycle. So we're interested in which groups are doing what in this system. And so this is just a simple relative abundance plot of who is there, who actually exists in this system. And you can see it is primarily dominated by sulfuri hydrogenebium, but also it's just a very simple, very small, very simple community, which is kind of nice when you're a bioinformatician. Makes things easier. And so we're interested in knowing which of these organisms are doing which of these conversions. And so I'm going to So in each of those genomes from each of those organisms, I looked for the enzymes involved, or the genes encoding the enzymes involved in these conversions. And so here, sulfuri hydrogenebium is a sulfide oxidizing organism. It kind of moves, everything in orange is going to be going in the oxidative direction, and everything in blue, which I'll show in a second, will be going in the reductive direction. So I'm actually going to flip through these very fast because we don't need to talk about every one, but you'll just get an idea that I did this to every single population of those highly abundant ones. So here we have the main populations, and you have which organisms are doing what. And then finally, we'll get to the summary slide. And this shows that these are the organisms responsible for the oxidative direction of the sulfur cycle, whereas these organisms are responsible for the reduction direction of the sulfur cycle. So that, as a microbial ecologist, that was very satisfying because it's like, wow, okay, that was pretty simple. You have these organisms doing this, these organisms doing this, and usually we're dealing with very complex communities. But then we took it a step further, and we saw that actually there's evidence of severe viral pressure, again, in this population, which probably can be said for any community, anywhere, ever. It's just not enough people are thinking about this. And so we actually recovered putative viral sequences that were associated with each of these genomes. And we looked more closely at it in particular, you'll notice here, sulfuri hydrogenebium, that highly dominant population, and another dominant, they have 35 viral sequences and 15 viral sequences respectively. So that's these two groups in the pie chart, the blue and the green. And what we noticed actually just a few days ago, which is really cool, is that within those viral genomes associated with those, they actually have biofilm formation genes. And so it's really cool to think about this network, this community, as actually a system of viruses, or a community of viruses that's actually promoting the stickiness, the stick-togetherness of that microbial community, because obviously that would be beneficial to the viruses as well, among many, many other things that the viruses can also encode. So this was a really cool finding that we're kind of excited about and working on a paper now. So yeah, in summary, you have these sort of gradient systems, these simple microbial communities existing across temperature and sulfidic gradient, sulfide to oxygen gradients. There's different organisms responsible for different parts of the sulfur cycle, and then there's extreme amount of viral pressure and even metabolic supplementation for certain aspects of the community. So with that, I'll just conclude that some broad level conclusions. Microbes play important roles in sulfur and carbon transformations in suboxic high temperature systems. Microorganisms that process sulfur and methane in suboxic environments have deep evolutionary roots. Evolutionary histories of the enzymes involving those processes are ancient and also subject to horizontal transfer events. And finally, viruses are very much overlooked for their roles in governing carbon and sulfur biogeochemistry. And so this is kind of what we're really attacking. Our research question now is well, how much do they actually affect that? So with that, I'll take any questions you might have.
Thanks. That was really fascinating. So this question about biofilm forming genes that are present in the viruses, and obviously as well as in the microbial populations.
That's correct.
Is there any way of parsing which of these genes are being expressed in actually forming the biofilm? Did the viruses actually play a role in that, or is that just they are just there in the environment?
That's a great question. There is a way to do that. We do not currently have that data, but that's what we're after, actually. So we've submitted 2 proposals, and we have a couple more upcoming that we're going to submit, but specifically doing transcriptomics and proteomics to kind of link the genes to the expression of those genes to then the actual proteins that make up those biofilm.
Hello? So that's super great talk. I really enjoyed that. So in your deep hole chemistry, you have this red arrow for your hydrogen sulfide, implying that it's magmatic hydrogen sulfide as an initiator. And yet you clearly have arrows that are basically doing sulfate reduction or sulfite reduction back to sulfide. So I presume that, and it's all imposed on this thermal gradient and chemo gradient. So it interests me to think about whether or not the activity of sulfide was high enough that you might have a gradient in sulfide precipitation along with these others. And if that were the case, it seems like sulfur isotope analysis could be a really fascinating complement to this study because, you know, in some ways you could lock up some of these pathways in sulfide mineralogy if the geochemistry permits. Is that?
Yeah, I think you're absolutely right. I think that's great. And one thing we're proposing, we haven't directly proposed for sulfur isotopes, but we are planning to look at clumped isotopes of methane because we're also interested in methane cycling through these systems. There's also a lot of methane in these vents. And we want to go back to Washburn Hot Springs and back to the hydrothermal vents in the lake and do isotopic work. We're working with, we are proposing to work with John Eiler at Caltech to do that kind of stuff. So I would hope, you know, that's always my goal is not to just rely on the bioinformatics and the microbial ecology or the microbiology data, but also to pursue that beyond to the level of really understanding what's going on with the geochemistry there. But I mean, you're absolutely right that there is definitely sulfur conversions going from different types of process, abiotic, biotic processes in different directions. And spatially, yeah, exactly. And I think one of the most interesting aspects of that sulfur cycle is that the elemental sulfur is the thing that precipitates either abiotically or biotically. And so that is the solid that sticks around. And so, we showed that elemental analysis, and those streamers are actually capturing elemental sulfur. So then they have a source of reductant or oxidant, depending on the organism, which direction it wants to go with that for that metabolism. And they just hold on to it in the community. So that's, yeah. And then if you think about, then the viruses are supplementing biofilm formation that's enhancing the holding on to of elemental sulfur. That's another interesting aspect.
So your hypothesis for prophase induction involves physiological responses, biofilm formation. Have you any hypothesis about what can be the environmental conditions that can trigger prophase induction?
You know, I don't know that I have a great answer to that particular question. I have a related comment. So first of all, we're collaborating with a viral ecologist in Olivia Nigro in Hawaii Pacific University, and we're kind of leaning on her to address those exact questions. But one thing with respect to the relationship between the viruses, the environment, and the microbial community that we find very interesting is when you have that level, when you have this level of viral pressure on a unified microbial genome, what does that actually do to that microorganism in terms of evolution? And we are hypothesizing that it actually puts so much pressure that this is necessarily a very fast evolving organism, because it has to change its genome very rapidly in response to viral pressure and also the extreme conditions of the vent. And so we actually see that when we analyze closely this genome, we see evidence of that, of this genome not being stable, but kind of being in flux. And so this is a question, it's very hard to get at this because an influx genome means a redundant, potentially contaminated genome, like we were talking about before. And so I think that I hope that a new era, wave of thinking in metagenomics and microbial communities comes in with this as microorganisms in natural environments, ecologically relevant microorganisms, do not exist the same way they do in cultures and laboratories. And the genomes actually change with respect to viral pressure. So I'm sorry, I realize I didn't fully, really address your exact question, but that is a question I'm going to rely on a collaborator to help me address. So thanks.
You talk about, I have two questions. You talk about elemental sulfur. Are you just going off the EDX data for that, or do you have any other observations to show that it's elemental
We
sulfur?
do have other observations. It's EDX and then also electron microscopy where we see geometrical shapes that are known to be elemental sulfur in these habitats. And when you have super hot hydrogen sulfide that gets really cold really fast in an oxygenated environment, it kind of...
Yeah, I just published on a paper showing sulfur lobus, sulfur inside cells using Raman spectroscopy. So we
cool.
can talk about that later.
Oh yeah, absolutely.
And a general question, the debate on Asgard archaeota has been really interesting as
Sure.
far as its state of play within metagenomics at the moment. I was wondering what your thoughts were on that debate and, shall we say, some of the kind of dark corners of metagenomics that have been illuminated by that debate.
Yeah, it's funny when we were trying to publish it, when our paper in Nature Micro was in review, and we were talking about that core archaeota. One of the we built something like 15 phylogenetic trees to try and really get at the evolutionary history of this organism based on different methods and different sets of genes used to compare deduced protein sequences. And one thing that we actually realized in several of our trees was that the core archaeota is more closely related to eukarya than Asgard. And so we were really careful because one of our reviewers was obviously an Asgard person. And they really did not like that finding. I don't know. So we just know that they were very promotional of the Asgard idea and they were very critical of our trees. But like I mentioned, Dr. Mincer Delakic at MSU, he is a wizard at phylogenomics and bioinformatics. And so any question that those reviewers were asking, he was able to just come right back with above and beyond responses. And so the reviewer ultimately agreed with our analysis. Now, we have other trees that showed Asgard is closer to eukarya. And this is another active research question that we have is really like trying to get more sequence information from these organisms and really look at that. Now, one thing, it's the Asgard eukarya hypothesis is not simply based on phylogenomic history or evolutionary history and close relatedness. It's also about having genes that encode eukaryote-like enzymes, which the Asgards have evidence of, and core archaeota does not have as much evidence of that. And so, yeah, we have like maybe a few challenges to that idea of the Asgard eukaryote story, but
I mean, if you take EF2 out of
yeah.
the whole debate, then it shifts so much.
Yeah. Yeah. Yes, definitely. Yeah, definitely.
We have time for one more.
Okay.
This is a somewhat naive question, but how much genomic variation do you have to have before you have a separate species? Just looking at this, if you have so much viral pressure generating so much apparent evolution on such a local scale, how do you know that what you're calling sulfur hydrogen or whatever it is in one area of Yellowstone is really the same species as something similar in another hot spring?
Yeah. So for one, let's say that you have your single, I'm doing this purposely with quote marks, single copy genes, which we know from cultures, microbial genomes are supposed to only have one copy of each of those single copy genes in their genome. And so that's how we estimate redundancy if you find multiple copies of those. So in this population, we did indeed find multiple copies, and we have a redundant genome. But we're able to actually look at the separate different copies of those and realize that they all belong, they're all related to sulfuri hydrogenebium. They're just not so closely related that they actually assemble together during the metagenomic assembly part of the step. So they actually are two different, they're two divergent, and so they end up on different scaffolds in the assembly. So what that means is they still retain the same tetranucleotide frequency, which is indicative of a constrained microbial population. And they are all related to that lineage of organisms, like at the genus or maybe family level. And so even if, it's like, call it what you will. It's the way I see it is there are some, organisms that are currently, as we speak, in evolutionary transitions. And there are others that are nice and constrained and they're happy in their environment. There's some that are actively adapting. And so what would it look like if you looked at a metagenome, and in that metagenome you had a population of organisms that was experiencing a lot of pressure and actively adapting to that environment? And I think it would look like sulfuri hydrogenebium. So hopefully that kind of answers your question.
Awesome. Well, if you have more questions for Luke, he's available now through lunch, and you're all welcome to join us for lunch if you have time. And yeah, let's thank Luke one more time. Thank you.
Thank you.